Broadcast is now starting. All attendees are in listen-only mode.
So hi all and welcome to today's ACTA webinar on the FDA guidance for adaptive designs for clinical trials of drugs and biologics. I'm delighted to be joined today by Scott Berry and Julie Marsh from our ACTA Statistics Special Interest Group STING. Scott will be presenting today, and Scott is President and a Senior Statistical Scientist at Berry Consultants. Scott's been involved in the designs of hundreds of Bayesian adaptive clinical trials of pharmaceuticals and medical devices and has become an opinion leader in the field of Bayesian adaptive clinical trials. We're delighted to have him joining us all the way from the US today. Welcome, Scott.
Thank you, Madeline. Happy to be here. Shall I start?
Yep, we're all good to go. Thanks.
Okay. All right. Thanks, Madeline. So I am going to share. I turned my picture off here just so the internet works well and fluid here. So thank you for the invitation. This is a wonderful thing to be able to talk about. The FDA, the US FDA guidance. So I will talk a little bit about it. I'll try to hit the big picture aspect of it, and then I'm even going to walk through several sections of it in pieces that I've highlighted. Things I think that are either missing or interesting in its inclusion, interesting in what they say. I hope this feels like a discussion on the setting of the various topics. I'll try to give my viewpoint on this. All right. So as you can see, the draft guidance, which came out in September 2018, September last year. I'll say a little bit about what's there. I'll dive into several sections of it. I may show some aspects of an adaptive design report. The document makes reference to such a thing. I'll try to take a look from, say, 15 years ago. I personally have been doing this about 20 years, and we started off doing Bayesian statistics, adaptive trials. So it's been interesting to see the evolution of what's happened here, the evolution of the FDA, things that maybe 15 years ago I would have been pleasantly shocked to see come out of the FDA. I think you'll find that I'm very, very positive on this document for many reasons. So a little bit of that as a big picture. So what I'm going to give is my viewpoint on this. It's the viewpoint of somebody who designs adaptive trials, typically Bayesian, in drugs, devices, for pharma, for government-sponsored trials, a range of different kinds of trials. So what's in the guidance? I want to move this thing so I can see my own slide here. So first of all, it's very positive. It's not restrictive. The 2010 FDA draft guidance that came out on adaptive trials was deemed by most to be negative. I didn't find it to be negative because it wasn't restrictive. There was nothing it said, there wasn't anything it said you can't do this. It was cautious. It used terms like not well understood. And I think it had a difficult time getting people over the top in that. And those of us that were doing it, it didn't hinder us, but it wasn't deemed to be a positive document necessarily. And I think people were looking for an invitation more than anything else. So the document talks about why adaptive trials. It gives some motivation. It does give some why not. It does a lot on what are adaptive things that could be done as a non-exhaustive set of potential adaptations. It talks a lot about how, whether it's how to submit it, what to submit. The how part around it, and consistent with the previous guidance and this guidance, the FDA is concerned with operational bias, leakage of information, the role of the DSMB and adaptive design committee. So there's a lot about how. And I think it gives you a window into the FDA's thoughts on adaptive trials, which is positive. So first of all, the existence of this document is a positive thing. And so everything else within that set, that's positive. I've highlighted multiple comments in the guidance itself. It's a reasonably short read if you haven't read the document yet. I thought that on this, to highlight this, in the document it says, in short, designing a clinical trial is a complex process, and it is not the intent of this guidance to require or restrict the use of adaptive designs in general or specific settings. I think it's a really important thing that this doesn't restrict the use of adaptive designs anywhere, in any specific settings or in general. And to me, that, no, I view this differently. That to me is a complete invitation to use them everywhere. But it certainly is not restrictive, and it says this is not restrictive. So the outline of this document itself is pretty interesting. So I thought I would walk through that. So there's an introduction of what is the scope of the document, a description of and motivation for adaptive trials. And first of all, there's a definition. Within the description and motivation, there's a definition. And a lot of people argue about the definitions of adaptive trials. And I think this one's a pretty good one. For the purposes of this guidance, an adaptive design is defined as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. So an important part of this is prospectively planned. Running a trial, throwing the data to the DSMB and letting it redesign your trial is not an adaptive trial by definition. We get a lot of people that come to us that want to do that. Oh, we'll give it to smart people and they'll do the right thing. That's not an adaptive trial. So that from the get-go, that's a really positive thing, I think, from the FDA. It goes through important concepts. It gives definitions around adaptive trials. There's not too much there. There's a little bit about comparative and non-comparative, and I'll say something about that. Motivation and examples, limitations, and the good thing is I don't think the limitations are restrictive at all. In some sense, they're rather common sense. And then the decision to choose to adapt. It goes through principles for adaptive trials. And I think these are really four very important parts of adaptive, of the guidance document. And much of the important parts of the guidance are around controlling the chance of erroneous conclusions. It's actually a really interesting way in which they do this. I think they didn't want the document to be overly type 1 error. So they talk about erroneous conclusions. In some settings, it's not clear if something's actually a type 1 error. So it's an interesting use of that. But do we make mistakes? Can adaptive designs do a good job at not making mistakes, including type 2 errors? Estimating treatment effects. I'll say a little bit about that because that's something historically we haven't done a lot of. Is the estimate coming out of an adaptive trial biased? It probably is, but to what extent is it biased? Trial planning. There's all sections on that. And then maintaining trial conduct and integrity. And this is something I think the FDA is really concerned with. Because if things go wrong, you don't know it goes wrong, and it's hard to tell. It's hard for the FDA to judge it, and they don't want to discount information that comes from trials. Adaptive designs based on non-comparative data, and in the terminology of this, they didn't want to use blinded and unblinded. It's really what it means is this is a blinded analysis. They think that masking and blinding is confusing in other settings. So a non-comparative data, you're not comparing arms in the interims. You're looking at event rates, measures of variability, perhaps to change sample size as an example. And then much of the document is adaptive designs based on comparative data, unblinded analyses of effect sizes that drive adaptations in trials. The adaptive designs based on comparative data, these are the bullets that describe the different adaptations. And I think they do a really nice job of it. They talk about group sequential designs. More generally, adaptations to sample size, adaptations to the patient population rather than sample size or which arms are given or treatments are given. Who do we treat? You might do enrichment, so that's a whole classification of adaptations. Adaptations on different treatment arms. Patient allocation, response adaptive randomization is an example of that, stopping arms. Adaptations to endpoint selection. It's a much smaller section. I think these are rarely done, and there isn't much in the guidance document about them. And it may be one point where they're a little bit cautious on that. Multiple adaptations to multiple design features. What's really nice is that this exists. It talks about combining 4 or 5 of the things above, if you wanted to do that in a design. And it doesn't say, it could say, for example, oh, you should be really cautious about combining multiple adaptations together in trials. It's typical regulatory speak, and there's very little of that in a setting. So it's, well, if you're doing multiple things, here's what you should think about. It doesn't say, oh, be cautious, we don't think you should do that. Then it has special considerations and topics, and this is where topics like Bayesian adaptive designs. And of course, from my perspective, the fact that this is part of the guidance document, it says we don't really care what quantitative measures you use. It's not an important part. Bayesian adaptive designs are adaptive designs. Specifically, design planning, a little bit different when you're counting events, perhaps, than patients. They talk about using surrogate and intermediate markers in adaptive trials. Again, the fact that it talks about this, gives guidance around that, is very positive. What happens to secondary endpoints, safety considerations, adaptive designs in early phase exploratory trials. Essentially, this document is mostly about phase 3 pivotal trials. And I think it's consistent with almost everybody's view at the FDA that early phase trials, yes, be adaptive, get better information. We're fully on board. You worry less about erroneous conclusions. You worry about them, but it's not quite as strict. And so much of the document is about phase 3 trials. They talk about unplanned design changes. You look at the data, you see an effect, and you do something that wasn't planned. This is where they're quite negative on that. If you want to find negative parts in the document, you'll find that, which I tend to agree with. In some settings, it can't be helped, but in many settings, it's inappropriate. And then what if external to the trial things come about and you want to make changes? They have a section that talks about that. And then maintaining trial integrity. And that's about operational bias, this strange a term that was all over the first guidance. And I'm not sure they actually say operational bias. They talk about trial integrity within this document. Regulatory considerations, interacting with the FDA, documentation prior to the adaptive trial. That's a big thing for them, this prospective aspect. What's being done was planned to be done. And then what do you do when it's over? How do you report the results of it? And then a set of references within the document. All right, so I thought at this point I would walk through and show, I'm going to come back to this PowerPoint, but I thought I would show the document itself and walk through various pieces of the guidance that I've highlighted, that I find there's a few things I disagree with. There's a few areas of things that I think have been left out within that section, a few things that I really liked. And I've tried to do them a little bit by color. I'll skip some of these comments in there. Here again are their definitions within the document. And let's go down to, sorry for the slowness of this. Here's a comment I actually disagree with, and I think it's inconsistent with the rest of their guidance. Adaptive designs require certain analytical methods to avoid increasing the chance of erroneous conclusion and introducing bias in estimates. So I don't think adaptive designs require analytical methods. It's nice if you have them. And then, of course, the next sentence is, for complex adaptive designs, such methods may not be readily available. The typical definition of a complex adaptive design is one where there are no analytical methods. The only way to understand the operating characteristics of the design is through simulation. They're incredibly inviting of that in this document. So I think that sentence by itself, if somebody pulled that out and showed that adaptive designs require certain analytical methods to avoid type 1 error inflation, I think that would be misconstrued from the general document. So it was a place within the document of the limitations. The others, I think, are fairly straightforward that most people would agree with. I showed you this about this document is not restrictive. Here are the 4 parts that I showed that I think that much of the document describes these 4 pieces. Erroneous conclusions, understanding the treatment effect estimates, details of the design is pre-specified. What does that mean? Adaptive design report, pre-specification, and then running of the trial and the trial integrity. I think that part is really important, that these are 4 major things the FDA is looking for when you bring them an adaptive design. And then it goes into these 4 pieces, it goes into the details of them. I found this section interesting, and there was no mention of this. This was an area where I thought it was interesting that something was missing, is that they talk about the potential for inflating type 1 error. And they start off with erroneous conclusions, and they start off with the usual group sequential of multiple chances for success inflates type 1 error if you don't adequately adjust. A really nice intro to this. But it gives the sense that adaptations inflate. There are a number of adaptations that actually deflate type 1 error. They do mention later that that can happen. But it has this overwhelming thing in the industry that looking at data is bad, it inflates type 1 error, and boy, that's a really bad thing. And I think by not mentioning that some adaptations have the opposite effect, futility analyses deflate type 1 error. Response adaptive randomization, depending on how it's done, can deflate type 1 error. So that was missing. This when I go to my section on 15 years ago, this is a section that we tried multiple times, say 15 years ago, to get phase 3 trials accepted based on do not disturb, okay? Phase 3 trials accepted based on proof of control of type 1 error through simulation. It was difficult. It was new to the agency. They generally required analytical control. To have this in a guidance that it may be necessary to use simulations to evaluate the chance of erroneous conclusions is monumental. It changes the whole landscape of adaptive trials from things that had to be simple enough to do analytical calculations to those that can be incredibly complicated we evaluate through simulation. It's a large part section 6.A. But the fact that this is here is really cool. I put this in yellow that they want to understand the extent of bias in estimates should be evaluated. I think that's a positive. I think it's a good thing. It's easy to do in simulations, but it's something we historically haven't done much of. We've simulated many trials, proof of type 1 error. FDA trials resulted in drugs being approved, devices being approved. And this is not something we've spent much time doing. We've had whole adaptive design documents that never address this at all. So I think this is positive, but it was something that was new to us as being people who do this quite a bit. And I think it's good. Trial planning, and this is one of those in green here. One of these sentences, is it one sentence that I think is really important and has a number of underlying things to it. It says, finally, complete pre-specification can motivate careful planning at the design stage. So by pre-specifying everything that happens, you go through a level of planning that typically is not done. You worry about all kinds of things, you build a better design, you reduce, and this is what they say now, reduce unnecessary sponsor access to comparative interim data. So a lazy adaptive trial requires humans to drive it. A pre-specified design is built, it has automatic moving parts, it can be run by a machine. I like to use the analogy that the design is now on automatic pilot. The adaptations, the randomization, everything is so pre-specified from the models, the thresholds, the timing, everything, that it now runs by machine. Humans should watch it like a DMC, but they're not driving it. Sponsors don't need access to data because they've made all the decisions ahead of time. They've thoroughly planned it out, and it takes away the need to see the data, get access to it, because you've actually made all the decisions as though you could see it through the design. And then it continues, and help ensure that the DMC, if involved in implementing the adaptive design, effectively focus is on its primary responsibilities of maintaining patient safety and trial integrity. They don't have to redesign the trial, do things they're not supposed to be doing. They can focus on safety and trial integrity. So I think it's a really powerful sentence on the benefits of well done pre-specified adaptive trials. There are a number of sections now on maintaining trial conduct and integrity, and they're very much trying to keep clinical individuals, peoples at sites, sponsors from seeing the data as it goes, or the information leakage that comes out that could potentially affect the trial results. And they give an example where perceived negative data might mean decreases enrollment rate, decreases adherence, decreases data availability, missing data, and affects the scientific integrity of the trial. So it's a really important part from the FDA perspective of trial conduct and integrity, and maybe even certain aspects of the design being designed differently so that stuff doesn't come out that could affect the trial. And they give some examples of that. So planning for an adaptive design trial should include a consideration of possible sources and consequences of trial conduct issues and plans to avoid these issues. Think about it before you're in the middle of the trial and now it becomes a concern. This is the section on unblinded analyses. It's really, I'm sorry, blinded analyses. It's really quite a short section. And then they get into adaptive designs on comparative data. You're looking at unblinded data, or the algorithm is looking at unblinded data. Here's another section that I don't think is entirely true, and again brings out this idea that adaptations based on comparative data, looking at the effect size of the arms, generally do directly increase the type 1 error probability and induce bias in treatment effect estimates. It doesn't say looking at the data causes that, but the adaptations taken. Now again, not all of them. And it says generally do, so I'm okay with this, but again, I thought that was an area where it almost feeds this notion that adaptations and looking can be bad. I don't think it was intended to be that way, but sometimes you pull sections out and they look like that. Here's a really nice part this is the adaptive designs based on comparative data. In that section, it ends with stopping or adaptations rules can be specified on a variety of different scales, such as the estimate of treatment effect, fixed sample p-values, conditional probability of trial success, Bayesian posterior probability that the drug is effective, or Bayesian predictive probability of trial success. The choice of scale is relatively unimportant as long as the operating characteristics of the design are adequately evaluated. They're not going to tell you to be Bayesian, be frequentist, to be a likelihoodist. As long as it's pre-specified, the choice of scale is unimportant. It's been simulated, and the design has good design characteristics. It's a really important part. They're not going to say, we're not Bayesian, we're not this. They're for good designs. A whole section on group sequential designs. They go through it, and I think it's pretty straightforward, not a whole lot of new aspects of that. There's a really interesting section on here, and we've been involved with this quite a bit. We like futility rules in trials. You don't want to continue trials that are going to fail. Continue to put people in the trial, spend time and resources and science on trials that are doomed for failure. So if you have a binding futility rule in the trial that if something happens, you stop the trial, that deflates type 1 error. So what it's saying is binding futility rules can provide some advantages in efficacy analyses. For example, a relaxed threshold for a determination of efficacy. So if I say I'm going to stop the trial when the following conditions happen, that deflates type 1 error to the point that I can raise the threshold for, think about an alpha for a p-value. I can raise alpha because I've deflated type 1 error. The raising of alpha increases it and gets it where I need to be. So it doesn't say you can't do this. Now, most of my interactions with the FDA, they don't like this to happen because they don't believe futility rules are binding. They worry DMC may recommend against them, it won't happen. Now you've bought back alpha in something that doesn't actually happen. So what they're saying is, if you're really going to do this, the FDA considers anything that hits that a failed trial, rightfully so. But it doesn't say you can't do it. So that was a really interesting part of the document. I was surprised at how open they were to that. Though we find most teams ask you to simulate any trial without futility rules to understand what the characteristics would be. They don't say anything about overrun and group sequential designs. You do interims based on some number of subjects passing the final threshold, and you have a big chunk of patients that aren't there yet. What happens to those? Is the primary analysis before them or after them? And I'm okay with it, but this is something that is really kind of vague in what people do. Okay, adaptations on sample size. A really important part of this, hidden in the paragraph on line 544, is that it is also critical that the adaptation rule and analysis plan are followed. This is now, I would say it's more strongly. If you say this is the design, it's the protocol. You said you're going to do it. It's critically important you do as you say you are going to do, and you follow the design so that we understand the results when they come out, the characteristics and the integrity. If you start doing things willy-nilly or don't do what you say, it may lead to a trial design that's uninterpretable. And so these are not guidelines or maybes. It's critical the adaptive rule and analysis plan are followed. Again, they come back to this idea of limiting personnel who has the knowledge in two ways. One, of the data itself, but also in some ways the details of the thresholds and the models and the design itself might be held back so that there can't be reverse engineering or back calculation that could lead to trial integrity. So they're very okay with the protocol not having the detail. It's in an adaptive design report that has limited availability, so that it avoids this issue of people seeing, aha, the trial's still running, that means X, and back calculating what happens. Adaptive enrichment. This didn't come out very well here, but there's an issue in adaptive enrichment where you might have three types of patients, and you're worried that the drug is ineffective in type A, but it's effective in B and C. You hope the design drops A and continues in B and C. If the design is successful in, say, A and B, and in truth, the drug works in B, but not A, is that a type 1 error? So you'll get different people to say different things. I say absolutely not. You approved a drug in a population where people benefit. It's a type 1 error when nobody benefits. Every drug ever approved have been approved for people that it doesn't benefit. It's the nature of heterogeneity. And so now in a design like this, when you explicitly address treatment heterogeneity, are there null hypotheses that aren't part of other designs? In a fixed trial without enrichment, we don't ask if we made that mistake. So when we do enrichment, we shouldn't ask that question there either. It ignores that issue. It's a complicated issue, and we've seen differential treatment at the FDA and there's nothing in the document about what actually a null hypothesis is. All right, I need to probably speed this up a little bit. It says seamless designs can be considered. Follow the principles of adaptive trial. Seamless phase 2-3. Select the dose, continue on to phase 3, and use the data from phase 2 in the confirmatory part. This is huge. Absolutely huge. And again, 15 years ago, this was relatively rare. And now it's, yeah, it's inviting. It talks about platform trials, adaptive platform trials as potential adaptations. It refers to other places, and this document is too small to deal with this whole development, but at least it recognizes it as a reasonable adaptation. They get to patient allocation, so response adaptive randomization. Talks about different ways of doing that. It says, here's where it does address a little bit this point I've said probably too much about. Response adaptive randomization alone does not generally increase the type 1 error rate probability of a trial when used with appropriate statistical analysis techniques. So response adaptive randomization may not inflate type 1 error, which in many circumstances it doesn't. It actually deflates it. Now that the other part I'll spend is talking a little bit about the simulation part. Clinical trial simulations will often be necessary to evaluate the trial design. And it's incredible that it doesn't say we don't accept simulation control of type 1 error. People for years have said the FDA doesn't accept it when we knew they did. And now they've actually gone out and they're inviting it. And they have a whole section on simulations, the importance of them, and using them for control of type 1 error. There's a section where it even talks about, while it is impossible to simulate every scenario compatible with the null hypothesis, it may be possible to determine a limited set of scenarios that adequately represent the plausible range of potential false positives. We can accept a finite set of null hypotheses simulated that demonstrate control of type 1 error. You can't simulate the infinite space, and we don't need you to. It's an enormous thing the FDA is laying out, and they even talk about how to create a grid to do this, to submit it. Incredibly welcoming of this idea. It also brings up the idea that, hey, and when the trial's over, you can actually do post-trial simulation to demonstrate for the data that occurred, we have control of type 1 error. So if people are still at all worried about it, post-trial simulation for the accrual rate and all the other components of it can be checked, which is a really important and innovative concept. Talks about sample sizes to control type 1 error within that setting, but it doesn't dictate some number, but give some guidance on that. Okay, Bayesian adaptive designs, it's positive on this. The one negative is it says it will often be advisable to use conjugate priors or computational less burdensome Bayesian estimation techniques. I sort of feel like it's 2019, MCMC is incredibly well understood. We use it all the time, that to push somebody to use it when it may not be the right thing, I wasn't in love with that sentence. The comprehensive discussion of Bayesian is outside the scope of this. I hope that means they come out with a Bayesian guidance. The Center for Devices has a Bayesian guidance, and again, that's quite welcoming. They talk about doing adaptations on biomarkers. Again, yes, do them. It may not be the primary endpoint, but you can do adaptations on early endpoints. It doesn't have to be a surrogate to do that. So very positive, early phase, very positive. They don't want unplanned design changes. Now they recognize things happen and sometimes you have to do something, but that's something they're quite cautious about. External data, and then there's a lot on trial integrity. There's one last section I'll put out here, and it's a new color. And it's something that I think many people don't think the FDA will accept, and we've seen them be reasonably accepting that there can be sponsor information about the results in the trial. There can be limiting unblinding at a sponsor. They have to finance the trial. They want to limit it, a small number, people that are not at a clinical site. But in here, they say, we don't think you should, and you don't, it's not a good idea, but there are specific situations where limited access for specific sponsor personnel may be justified. And so this is definitely something that can be done during the course of the trial. And we get a lot of people that don't think that's something that can happen. Okay, pre-specification is really important. Here's where they talk about, instead of having a design that says, okay, now you need 313, put it in discretized buckets so you can't reverse engineer effects, things like that. Another part of this is that they talk about a specific committee to implement the adaptive design that may not be the DMC. Let the DMC worry about safety and let perhaps statisticians do the adaptive design. It's part of the protocol. They can do that. Now, it's not uncommon that's done within a DMC, but they talk about this group needed to run the protocol. All right, they talk about what to submit, submitting code, submitting an adaptive design report in the pieces, but I'll not show you that part. We can talk more about an adaptive design. I'm happy to send these slides out. I gave you an example of an adaptive design report that I think touches everything the FDA says they want in the report that's sent to them. This is the ICECAP trial. It's hypothermic cooling for post-cardiac arrest. It's a NIH submission, so it was okay to send that document out. So I'll end and open it up to questions, but just say something about 15 years ago. Most of these documents are cautious, and the last one was perceived overly cautious. This is not. It doesn't say, for example, we prefer fixed trials and you need a strong rationale for an adaptive design. That's been in other documents before. It's not said here. And they said it's not restrictive. Very open to adaptation. They don't say we're open to phase 2, but not so much phase 3. Doesn't say that at all. It says that you can control type 1 error through simulation. They even give you guidance on how to do it. That was rare 15 years ago. Looking at this 15 years in the future, it would have been wow. That's pretty incredible. Frequently they say things like, oh, you shouldn't do more than 1 or 2 interims. We do 20 or 30. There's no reason why you only do 1 or 2. They never say that in the document. Again, a lot of stuff is positive by omission. And I'll just point out that if looking back 15 years ago, how forward thinking this is from the FDA and how positive it is, what are we going to see in 15 years from now? Pretty exciting. Okay, so I will stop there, and I think I turn it over to Julie, and happy to discussions, questions.
Thanks, Scott. You've run through an amazing amount of information in a quick time. I do have a list of questions from everyone, but lots of comments as well saying how they appreciate, the guidances which are coming out, in particular around being able to provide a report as well of what's needed and when. So maybe we can kick off initially with a question from Steve Webb when he said, can you explain how response adaptive randomization decreases the likelihood of the type 1 error?
Yeah, so sure, Steve. So think about a case where you have 3 doses and a placebo. And if you run a fixed trial, you have 3 doses, so you might adjust the alpha, and you have, say, a 5% chance of type 1 error. If you early in the trial you look, what happens in a type 1 error? The null is true, and all 3 doses are ineffective, but one has a random high and makes it look like that dose is good. Or if you have 3 experimental arms, it makes it look like that arm is good. When you do an interim, the arm that's doing the best gets more patients. So it's actually the exact opposite of what group sequential does. Group sequential, early stopping for success, when data's good, you decrease the sample size. That causes inflation. It's easier to get a random high. Response adaptive randomization puts more patients on the better arm. So if the null is true, you give it more opportunity to regress to its truth of ineffectiveness. It's like increasing the sample size for arms that are good, which is the exact opposite adaptation of stopping early when data are good. So it has the exact opposite that has. It deflates type 1 error. It doesn't work in perhaps a 2-arm trial. If it's A against its placebo, RAR favoring the arm decreases the placebo, and it might not change the relative effect size. But in multi-arm scenarios, it decreases type 1 error. If the response adaptive randomization puts more patients on better arms, which hopefully that's what it does.
Thanks, Scott. That's great. Something we struggle with a lot is the amount of statisticians these trials appear to need following these guidance, and whether we can really fund these out of the more investigator-led trials.
So, Julie, the concern is that the more effort to build them and the role of the statistician, the increased resources, that's the question / concern.
Yeah, and also the monitoring of the trial,
Yeah.
the idea of a separate committee for running the adaptations, in addition to the data safety and monitoring committee.
Yeah, so it is. My hope, of course, is that as we get better and better at this, that it's not really much of an increased cost. The simulation code that did it, that's just used to run the interims, and this data availability is better, and this is a much smaller cost. Hopefully, the efficiency gained overwhelms that additional cost, but I think as we're getting up and running and doing these, it is an additional cost. Most sponsors, and that's pharmaceutical, larger money, the gains of the efficiency outweigh that. Yes, in perhaps federally funded trials, grant funded trials, sponsor initiated trials, it may be hard to get those resources. It may be hard to do this well. And it's an issue also tied to the issue that we need more statisticians that do adaptive trials, that are experienced in them and do them well. But I think it is an issue as we move forward.
But the other thing that comes up,
I shouldn't pause and then interrupt. We find it's hard when you're submitting things to agencies to put a great deal of effort forward in the simulations when you don't know you're funded. So we've tried to work with the US NIH on the ability, and Berry Consultants actually has a grant with the NHLBI to work with groups on adaptive trials. And I think that work should be done when you know it's going to be funded. Let's improve the design. It should really be part of it. It's hard to put a ton of effort in when you don't even know if you're funded.
I think that's a really great point, Scott, that maybe we should, as a actor group, form what is sufficient in the simulations for grant applications to give people some idea of what to expect, because we often find that you put a basic simulation in and of course everybody can pick holes in it, but we're trying to provide a simplified situation early on to see if it's plausible for funding.
Yeah, and it's hard to be funded for work you did before. So to be able to have a 6-month run-in while you're getting your sites up and running, to really do it well, and that's part of the funding, would be great. But you're right, people have to recognize that when you submit it, you've submitted a smaller set, less simulations. You just don't have the time and effort.
I think the other major theme to come on to is trial integrity. I was particularly interested what you were saying about a separate group that runs the trial. Could you give a bit more detail around, what is the FDA looking for in terms of regulatory or minutes or independence? Is that a whole new source of bias that can come in?
Yeah, we've been involved in a number of these. We've been that committee sometimes.
Did we just cut out because the room hit 15 minutes?
So sometimes there's CROs involved with that aspect of it. So what they would like is a group independent from the sponsor who carry out the design. It's getting the data, running the models, checking the thresholds, resetting the IVRS, doing those components of it. They're separate from safety, they're separate from the other pieces.
The whole thing may have cut out.
Oh, can you hear me, Julie?
Yeah.
You can hear quite fine, Scott. Keep talking.
Okay. Thanks. So that's an important part of making sure it gets run well. They usually have a good bit of archiving all the interims, and then when the trial's over, you submit what the data look like at the interims, the decisions made, and they can see that the design was followed. They want to know that the design was followed during the course of that, and you've got all of the interim stuff archived in the setting. A lot of times it's statisticians. We have a charter for the group. It specifically says our charter is not safety. We're running the protocol. We're statisticians that running the protocol, and that's not our job. And I think it's the FDA's role of making sure this goes well. There have been times where we've designed trials where they've asked that groups other than us run them, that perhaps there's a bias in a group being involved in it. CROs do it, a number of groups do it.
Scott, it's Madeline here. Unfortunately, we've just lost Julie. Her internet connection's cut out.
Yeah.
We do have a couple more questions that have come in, so I might just pass these over to you now. We have a question here from Andrew Forbes. Just a general question. With the ASA push to abandon statistical significance and the dichotomization of p-values, what are the implications for designing adaptive or any other trials?
Boy, it's a great question. By the way, Madeline, it's a great example of being adaptive on the fly. Unplanned interims being done. We're doing it well. So Andrew, it's a great question. I actually think, and I sometimes feel bad about it, but the one area where we do p-values well, we pre-specify everything, is clinical trials. So a lot of this push, I think, are post hoc analyses, poor pre-specification, fishing expeditions, and then writing down P of 0.013. We don't typically have that issue in clinical trials. So I'm not so sure that this is the area that the ASA and people are the most worried about. Yes, there are people that still think p-values are poor measures of everything. I fall in that group, but we do it pretty well in clinical trials. If that goes away, I think that adaptive designs will always be there. It's going to get the answer more efficiently. It's going to waste patients less often, but we got to know what the goal is. And it might be a posterior probability, it could be a maximized utility, whatever it is, the adaptive design will do it better. So I don't think that affects it, even though it's certainly related to type 1 error and all of the aspects of p-values.
Thanks so much. Just a few minutes left, so we might just have time for one more question. We've got one here, another one from Steve Webb, just asking if you can comment on the issue of accuracy of estimation of size of treatment effect in adaptive trials and any methods to deal with the issue.
Yeah, so Steve, I gave you the example where we have 3 experimental arms and a control, and we might do response adaptive randomization. And one of the arms wins. There may be a bias in that estimate that the arm that wins has a positive bias. We may pick a subset of patients that in group A, this drug works. And we look through multiple things and we've controlled type 1 error, but the pure estimated effect within group A of that drug is higher than we believe it because of the cherry picking. We control the threshold, but that doesn't mean we necessarily change the estimate itself, the mean effect. So you can run the trial through the simulation and calculate the characteristics of those estimates. I'll push this past to the top of the hour. I could spend a lot of time on this. I think this is a great idea. In I-SPY 2, this happens. We have 8 subtypes of breast cancer with 10 potential signatures a drug could graduate in. We know the raw rate of pathologic complete response, that's the endpoint, in that subset it graduates in is biased. We simulate it, we know it, so we provide a model estimate of the effect, and we don't provide the raw data. And it's shocked the industry. Wait, what do you mean you give us a model estimate? What was out of the 64 patients in that subgroup, how many were PCRs? We don't want to give it because we know it's inflated high. The model estimate is adjusted for it. That's what you should know. It's really hard and it changes the industry, and perhaps we shouldn't be quite so dogmatic that, look, here's the observed data. Here's the estimated bias. The model estimates 36%, not 43% for that subgroup, and people getting used to that coming out of adaptive trials. So it's a really important thing as labels come out. The FDA is worried about the label. Usually they say to us, that's a good problem to have. You show the drug works and we'll figure out the label. That's a good place for us to be. And as we get more and more adaptive trials, we'll have to see how this all shakes out.
Hi, Scott. I'm aware that we've hit the hour. We need to round up pretty quick for people to get to move on with their day. That was quite a shocker to finish with, and the guidance you've given us throughout has been fantastic. I suppose the only last thing is this consultation still open? Are the FDA still looking for feedback?
Oh, I think the feedback time is officially closed, though I suspect they would take feedback.
Okay, so thanks from everybody for tuning in today, for sending great questions, and a huge thank you to Scott for giving us such clarity and support in adaptive designs in Australia. Have a great day, everyone.
Thanks, Julie. Bye.